Okay, so today's lecture is about the multiomics, means how we can use various omics, named as metabolomics, proteomics, genomics, and transcriptomics in our research to know, understand more about the disease, the gene expression, how protein is changed, and how different metabolites are changed during the disease, or if they are changing, how they can lead to the disease process, underlying cause, and those things. So main thing is to understand the use of multiomics in the research. That will be the learning objective. So multiomics comprises four things basically: genomics, transcriptomics, proteomics, and metabolomics. And it helps in comprehensive understanding of the complex relationship between various genes, proteins, metabolites, and how the change in expression of one thing can lead to the change in the expression of others. It allows researchers to analyze different molecules at the molecular level, that is gene, protein, RNA, all those things, and it provides a more holistic view of the complex biological processes. Omics data include genomics, mainly for DNA, transcriptomics to understand the change in RNA, proteomics in protein, and metabolomics in metabolites from various whatever we are eating or whatever the metabolites are there in the body. So integrated omics data can help in understanding the disease process better, how the genes, RNA, proteins, and other molecules are interacting with each other and how they can influence the biological process going on in our body. And this can also identify the relationship and pattern that might be missed during our normal routine procedures, like if we are doing RT-PCR, ELISA, Western. So there we are looking for the expression of a certain gene or protein. Not we are not looking the relationship between them or how they are interacting with each other. We can analyze the comparative data, right? Like in control versus disease process, whether this gene is overexpressed or underexpressed, or its expression is increased or decreased, but how it is related to the control, and if it is decreased or increased, how it will affect the biological process in the human or any organism. We are not doing that in our routine lab processes. So for that, we use metabolomics, multiomics, I mean. So multiomics can be applied to human disease, means if we want to understand any disease in more depth, to understand the intricate relationship between change in gene expression and how it will lead to change in a protein expression and how it can affect the change in the metabolomics of our various metabolites in the body. So all those things we can look into by using these omics, and this will help in understanding the disease process in a better way. And also it will help in investigating or delineating a specific target which is responsible for that metabolic changes or biological changes, and then we can target that with the drugs. So then personalized medicine means, if some, as I discussed in previous lecture also, that if there are some patients who are not responding to medication or the treatment, then we can compare them with the patients who are responding. That will tell us that what is the change in the gene expression or protein expression or metabolite expression is there in the patients who are not responding compared to the patients who are responding. So in that case, we can you target those change expression either in gene, protein, or metabolite to make them more responsive to the treatment which we are giving. That is called as personalized medicine. Means we can personalize the medical treatment for each and every individual by using the omics. Then drug discovery means, if we are working with the omics data and we find that there is a gene or protein which is highly expressed, and that gene or protein is responsible for the disease process, then we can use various softwares where we can find some small molecules or drugs which can be used to target that gene or protein. And then we will do the wet lab experiments to find that whether that drug or small molecule which we are predicting using the omics data set is really targeting that gene or not. If it is, then that will lead to a new drug discovery. Single cell analysis. So normally when we are doing our wet lab experiment like RT-PCR or Western or ELISA or immunostaining, these are the routine